When we look at the documentation of the cell cycle scoring function from the Sura package, you can see that it uses the sets of marker genes to predict the phase that a certain cell is in. So for each cell, it will check if it expresses these marker genes. So if a cell expresses most of the marker genes for the S phase, then it will predict that that cell is in the S phase. If a cell expresses most of the markers of the G2M phase, then it will predict that the cell is in that phase. And if a cell doesn't express any of the markers that are listed here, then the function will predict that the cell is in the G1 phase. So it will work on a Sura object, as you can see here, and I give him the two lists of names of marker genes for the two different phases of the cell cycle. The function will often throw a warning. It is saying here that certain marker genes are not present in our dataset, but this was to be expected because the cell cycle markers here are human genes and we are working in a mouse dataset. The cell cycle scoring function adds a number of columns to the metadata. The phase column defines for each cell the predicted phase of the cell cycle. So the first cell is believed to be in the G1 phase. The second cell is believed to be in the S phase of the cell cycle. Apart from the phase column, it also generates two columns with scores. So for each phase in the cell cycle, the score reflects how likely it is that the cell is in that particular phase. So for the first cell, the scores are low because the cell is thought to be in the G1 phase. For the second cell, you see that the score for the S phase is relatively high, and this is why it was predicted that this cell is in the S phase of the cell cycle. So we are still working with the copy of the Sura object, so we're going to add these three columns to the original Sura object. So we retrieve every column from the metadata in the copied Sura object, and in the original Sura object we create a column in the metadata with the same name. And then we can remove the copy of the Sura object. And now we can go through the entire Sura pipeline with our Sura objects, so we can find 2000 most variable genes. We scale the counts of the most variable genes. We perform the PCA. And then we create a PCA plot. We're going to color the cells according to the cell cycle phase they are believed to be in. And we're also going to ask to create 3 different PCA plots, one for every phase in the cell cycle. So these are the cells believed to be in the G1 phase, these are in the G2M phase, and these cells are in the S phase. What do you want to see on this plot? You want to see a similar shape for each phase of the cell cycle. So if the shapes of the clouds of points are similar, it means that the cell cycle is not a major contributor to the variability in the data. However, if the shapes of the clouds of points are very different, then it means that indeed the cell cycle is contributing a lot to the variability in the data, and it will probably affect the clustering. Many people prefer a UMAP plot over a PCA plot, so I'm going to run the UMAP. And I create a UMAP plot where I color every cell according to the phase of the cell cycle it's in. So here you look for clusters that consist completely of cells from one phase in the cell cycle. So you see that we have a few of these clusters here. If it's only a limited number, it's probably okay, but if you see many clusters that consist solely of cells in the same phase of the cell cycle, then you have an issue. It is to be expected in that case that cell cycle is driving the clustering, which is not always what you want. So depending on the biology that you are studying, you might want to get rid of that cell cycle effect. So if you're studying any biological processes where cell cycle plays an important role, of course you want to keep it in because it's the thing that you're studying. But if you're studying something that is unrelated to cell cycle, then you might want to get this cell cycle effect out of your data because it might obscure some of the real biological variability that you're interested in. So if you want to get rid of the cell cycle effect, you can scale your counts again. So we have already done this. We are going to go back a few steps in the Sura pipeline. We're going to scale the data again, but this time we are going to set the vars to regress argument to the two columns in the metadata that contain the scores for the different phases in the cell cycle. So the scaling will remove all the differences between the cells that are caused by having different scores for these two phases in the cell cycle. At this point, I'm only changing the counts for the 2000 most variable genes. If you want to do this for all the genes in the dataset, then you also have to set the features argument. You have to set it equal to the row names of the Sura object. So the row names are the gene names. You are retrieving the names of all the genes in the Sura object here, and you will correct the counts of all the genes in the Sura object. When the scaling is finished, you can run the PCA and the clustering again, but this time based on the corrected counts. So I repeat the PCA. I look for the nearest neighbors of every cell in my dataset. And then I cluster my cells based on these results using the same resolution as before. You see that he finds an additional cluster now, so the clustering has changed because the counts have changed. I want to make a UMAP, so I'm going to run the UMAP again. And then I create the same UMAP as before, as this one, but this time based on the corrected counts. And I put both of them next to each other so I can compare them. So it is essentially the same UMAP, it has just rotated a bit. This happens because UMAP has a bit of randomness inside, so it doesn't always give exactly the same results. And what you typically see is that when you do it multiple times on the same data set, you sometimes get results that are slightly rotated. So it's very much alike. But you do see that these clusters here that consisted completely of cells that are in the same phase of the cell cycle, that they have been integrated now here inside the other clusters. All variability that was caused by differences in cell cycle phase have now been removed from the dataset. So you get a slightly different result, and of course you will get slightly different clusters. Here also you see that it has changed a bit compared to the UMAP with the clustering that we have found in the basic Sura pipeline.